Okay, everybody, we're going to go ahead and get started. Thank you for your patience. Some people may be coming in still. That's okay. I want to introduce myself. My name is Meg Sewell, and I am the Director of Outreach and Recruitment and what they used to call education, but now we call it engagement. So I'm here to engage you in today's Participant Appreciation Day. We're so glad that you can join us. This is the first in-person Participant Appreciation Day that we've had since 2019, right before the COVID pandemic. So we're thrilled to be able to do this again in person. So, you know, I just want to say that everything that we know about Alzheimer's disease, every discovery, every clinical trial, every step forward for prevention or for treatment has been made because of people like you, quite simply. Every single bit of science that happens is because people like you are willing to show up and to volunteer their time, often year after year, and to show up like good, hardcore New Yorkers that we all are. You show up in horrible New York City traffic, in horrible freezing cold winters, during government shutdowns, during pauses in our funding. Nothing has kept you away, and we are deeply grateful for that. And yet, one of the biggest challenges in our field is that we simply don't have enough volunteers to participate in research. And that's not something that everybody knows. We often talk about how hard it is to get funding for studies and things like that, but nearly 80% of trials across different medical specialties have either been delayed or even stopped altogether simply because there aren't enough volunteers to be part of those studies and to answer those scientific questions. And as you know, Alzheimer's studies are among the hardest to enroll. So that makes your participation in our studies all the more meaningful. And I want to assure you that your participation is a huge part of why the field of Alzheimer's research is moving forward, and it is moving forward. And you're going to hear some more today from various people about what that's looking like. So again, just thank you from all of us here for your time and your commitment. And we're so happy you're here. We have a lot of things going on today, and I'm going to be here to walk you through each step of it. And the first step is going to be hearing from Dr. Mary Sano, the director of the ADRC, who's going to be here to give a talk. Yes, I'm sorry, it is. She's going to talk about preventing Alzheimer's disease and supporting brain health. So please welcome Dr. Mary Sano. Do you want to talk from there and I'll advance your slide?
Sure. Well, thank you, everyone. It is so exciting to see everyone here. Since 2019, really, an applause for yourselves. Thank you. Thank you all for coming. As you heard, I'm going to start us off with a little bit of our success, what we have actually accomplished in the last decade, which all of you have helped us with in different ways, right? And I'm going to talk about how it leads us to the next step in terms of prevention and in terms of brain health. So on the next slide, these are the things, whoops. These are the things that I'm going to talk about quickly. I'm going to tell you about how we diagnose and treat Alzheimer's disease. It's been updated since we met in 2019. The challenges to something we call secondary prevention. Many of you may be involved in some of those studies. I'm going to talk about what if it's not Alzheimer's disease, right? We want to keep the brain strong for all of the diseases. And then where does brain health fit in? How do we think about the kinds of lifestyle changes that might actually improve our overall brain health? Next slide. So if you remember, Alzheimer's disease is one of many dementias. And in fact, while I'm showing you that Alzheimer's may be the most common, probably people have several at one time. So we really know the complexity of the brain. And you're going to hear so much more about it when you hear from our panel and about our brain donation program. The next slide. So the big news, though, is we can now diagnose Alzheimer's disease. We no longer have to use that word, probable Alzheimer's disease. What this slide is showing you is that in 2010, we had an important publication that told us that we can image amyloid in the brain. And that can tell us, without someone having to die and get an autopsy, whether or not they have Alzheimer's disease. But as I just mentioned, there's so many other things that a person can have. This is only the tip of the iceberg. In the next slide, I'm going to show you the fact that we've even gotten better, because while those images are so perfectly telling us where the amyloid is, they're expensive. They require that we get something called a ligand to come to a center. It's very complex to get these images done. And if we think this is a common disease, we need a way to find out if everyone has it or if anyone has it. So we now have blood tests, and I want to show you how we're using these blood tests. Patients with cognitive impairment who are seeing their doctors, complaining about their memory or other cognition, or perhaps having a cognitive assessment that tells us that there's something wrong, they can take a blood test, and what we can do is determine whether they have high or low or intermediate probability for having amyloid. Right? And that tells us, if they have amyloid, that they are likely to have Alzheimer's disease in combination, or that is the likely cause of their cognitive impairment. So this is a huge step forward. Do you remember a time when we always talked about probable Alzheimer's disease? We're getting confidence with less probability and more certainty. Next slide. So this slide is a very interesting slide, and I'm going to ask you to look at it, and I'll come back to talk about it. What it shows you is that we believe the pathology of Alzheimer's disease, that's the red line of amyloid, and there's a line of synaptic dysfunction. Those are the nerves that aren't working and communicating very well together. Or tau, another protein. Those things seem, we thought, to occur before we see any cognitive problems. So a big challenge is to determine, when should we start measuring Alzheimer's disease? When should we start measuring that pathology? And in the next slide, I want you to see that we actually have discovered that even in very mild patients, or patients who have mild cognitive impairment and have Alzheimer's pathology, we've now been able to treat them. We have 2 drugs on the market. We're able to treat them to remove the amyloid and to see a clinical benefit. And what these graphs show you is that with this particular drug, lecanemab, what you see is there's a benefit on 4 different outcome measures. One is something called the CDR, and if you're participating in our studies, you've probably had the CDR, where we ask you and a family member or a friend about how you're doing, and we put it together to get a score. What we're seeing is that those people who have Alzheimer's disease actually do better on this drug. And we actually see the benefit on all of our outcome measures. So this is a real step forward. In the next slide, here's a second drug that was approved in 2023 that also has the same thing, right? It actually is able to show a clinical benefit. And in the next slide, what you see is we believe the clinical benefit comes from the removal of amyloid. So this is individuals who have amyloid in their brains, and the ones on the treatment, the amyloid is nearly completely removed. We've had other drugs that tried to remove amyloid, but they weren't quite so successful. But now we have ways to do this that not only are making a big difference, but they're also associated with having a clinical benefit. That's a step forward. Next slide. Now, there's more to know about these drugs, right? We're looking for new ways to deliver them. They're currently delivered through infusions, which can be burdensome. We're looking into ways to make them available by injection. And they do have side effects, and they're not for everyone. But you can't forget that it is a step forward. And we made that step forward because so many people helped us with this research. So now what's the next step forward? In the next slide, I want to talk about treating Alzheimer's disease, that's the pathology, to prevent cognitive loss and dementia. Can we do it? Should we do it? When should we do it? So many questions. So in the next slide, consider that everyone over 65, maybe it's younger or older, depending on whether you have a family history or a risk factor, gets the blood test and determines whether they have high, intermediate, or low probability of getting Alzheimer's disease. Well, if you had no symptoms, if you had no problems, and you were in that high probability, would you really want to go and treat if you didn't have any symptoms? We don't know the answer. But if I could tell you that treating means that you didn't get the symptoms for a longer time, or that you didn't get them as severely, that might be something you'd want to know. Not saying this is the right thing to do yet, but imagine that we might be able to figure out a way to stop the disease from causing the cognitive deficit. It's just a theory. We don't know. We have to find out. In the next slide. So I want to be sure that you understand what this direction of research is. We need to understand the full impact of pre-symptomatic amyloid positivity. What does that mean? It means some of you may actually be in some of our studies that are examining this. Our AHEAD study is a study in which people who have no cognitive complaints or symptoms, but do have high amyloid, they are being followed with one of these drugs to see whether or not it can prevent or delay the onset of symptoms. That's a very big step forward. Again, may not be for everyone, but we haven't had a new drug for more than 20 years. So this is a big deal. And we're really moving in this direction, not only of early treatment, but of prevention. So the other question is, what if it's not Alzheimer's? Right now, this is an important question because everyone, you saw that Alzheimer's disease was the most common one, but it might not even be the first one. There may be other diseases that occur first and the Alzheimer's comes later. In fact, we did a study, and we also participated in this study. It was a national study of individuals who had Medicare. And when they went to their specialist, this is long before we had these tests, we wanted to find out if they could be helpful. We said, well, if the doctor thinks it might be Alzheimer's disease, he can order this scan that Medicare will pay for that will tell us whether or not the amyloid is present or not present. Right? Now, these are specialists. These weren't just your internists. These were specialists in dementia. And basically, almost 40% of the people that they thought might have Alzheimer's disease did not have that pathology. So that leaves a lot of people out there who have something other than Alzheimer's disease, at least in the beginning. So in the next slide, I think for those people, we always say treat your treatable conditions, but we really need to know what's the best way for people who are worried, maybe concerned. How can we prevent cognitive impairment and dementia? Is there really such a thing as brain health that would maximize our cognitive function regardless of what our pathology is? So I want to tell you about the Pointer study. The Pointer study was announced last summer. It was a study of lifestyle interventions in which we had more than 2,000 individuals who were either assigned to being given information about healthy lifestyles or were actually engaged in activities on those healthy lifestyles, like physical activity, monitoring their diet, cognitive stimulation, social stimulation. And what we discovered is in these individuals, particularly those who were sedentary, did not engage in much activity, had a limited diet, had low social and cognitive activity, what we found is there was a benefit. So in this next slide, good. What we're seeing here is that now there's something very important to notice. These people are not demented, and so with practice, their scores get better, not down, but up. But what you're really seeing is that with the extra support and guidance, these people had an even bigger benefit on cognition. And what's really interesting is most of the cognitive benefit was in what we call executive function and processing speed, a little bit less so in memory. So this tells us that this healthy lifestyle does have a place in our senior life to maximize our cognitive status. It won't take the place of preventing a specific disease, but it definitely gives us an opportunity to get the most out of our cognition. So what are the takeaways from this pointer study, which was sort of an exciting result because it was done so carefully? We did find that enhancing lifestyle can improve cognition, and bigger effects were present when they had support, moral support, and people helping them do the work. The biggest challenge with lifestyle, and we all know this, is compliance. If you don't do it, you don't get the benefit. Even if you want to, unless you do it, you need the benefit. So staying engaged is really an important aspect to any of our lifestyle interventions. And if we think about it, it's a possibility that it's a treatment for all aspects of our taking care of ourselves, staying engaged, being compliant, following the orders that we've decided we want to follow. Okay, so I've sort of given you a lot of things to think about, and there's going to be a full panel of real experts here to answer questions about these and anything else that you have. But I wanted to just start by opening, showing you we've made progress and that we have a very specific direction that we're going in to make even more progress. So just to begin, Alzheimer's disease is a specific disease and we can now detect it. It even has a treatment, not for everyone, but for many. Research and focusing on Alzheimer's disease before we have symptoms is our next big hurdle. We want to see if we can prevent those symptoms from actually occurring. But it's also important to know that not everyone has Alzheimer's disease. And that's why we talk about ADRD, Alzheimer's disease and related disorders. There could be other reasons why we have this cognitive problem or dementia. And I also want to point out that lifestyle interventions, they really can make a difference. And we want to encourage both your participation in this research and a real attention to your health care at all aspects. So I'm going to stop there. Oh, next slide. I think I have my most important slide. You've seen this all day. We don't do anything except what we do together, right? And this is our team, and this is our team. And I really want to tell you that we're very excited to keep moving together on these really important journeys. So thank you.
All right. Thank you so much, Dr. Sano. So now we're going to take a minute to bring up to the stage the panel that's going to be our next talk. And if you have questions for Dr. Sano's presentation, just hold them a little bit because we're going to get to that a little bit later in the day. So can I ask Dr. Crary and Dr. Hedden, Dr. Pereira to come on up, and Dr. Nugrishel? Morning. And I'm going to pull up some slides. Hi. Yes. Also, we're going to ask our two research participants, Alex and Christiana, to join us on stage. So Anna, you're going first. So you can use this. This was just going.
Good morning. Can you hear me? Yeah, great. Thank you so much for the invitation and for all of you coming today. None of the work and none of the advancements that Dr. Seno has pointed out could be possible without the help of multiple research participants just like you. I'm a cognitive neurologist here, so I'll try to be brief so that you have time for questions and for a lively panel discussion as well. So those are my disclosures unrelated to what I'm talking today. So Alzheimer's disease is a devastating disorder. It's a progressive disorder, and many of you, it's not working.
A little hard because this doesn't go. There we go. Okay, instead of moving forward.
Better? Yeah, thank you. Yeah, so I was just saying that none of the work and advancements in the field that we have made so far could be possible without the help of each of you and multiple hundreds and thousands of participants who have helped us so far. We're deeply grateful to all you do and the generosity to help advance science for this generation and the future generation. Thank you. I'm a cognitive neurologist here at Mount Sinai. And as many of you probably have seen patients with Alzheimer's disease in your family members know the devastation of this disorder, which usually starts affecting memory in the hippocampal formation and then it slowly progresses to affect multiple aspects of cognition, including executive function, ability to plan, execute, emotions, thinking, and all those key abilities that make us who we are, human beings. It really steals the identity of people. So it's our mission to try to do better in preventing and treating those patients. Alzheimer's disease is a very complex disorder, and in the last couple of decades, we have started to understand a lot better about the biology of this disorder. I don't know if I have a pointer, but two key pathological hallmarks of the disease is amyloid plaques and neurofibrillary tangles that Dr. Crary, the neuropathologist here, may talk about. But there are many other aspects of this disease that we are learning and potentially be able to target. And you may end up having multiple medications, just like to treat cardiovascular disease, to ultimately be able to treat this complex disorder in a much more effective way. So thanks to participants just like you, we have been able in the last couple of decades to understand the temporal spatial progression of this disease. Through imaging, participants at high risk, especially those who have mutations who are deterministic to develop Alzheimer's disease, we know that the amyloid plaques start accumulating 15 years before any symptoms start. After that, about 7 to 10 years, you have hypometabolism, less metabolic activity in the brain. And 5 to 10 years before starting having symptoms, you're starting having structural atrophy. The brain starts shrinking. And for me as a neurologist, I can see that in the brain MRI. And then we have the tau PET that Dr. Hedden is going to talk about. When it turns positive in the neocortex, that's as close as you can get to symptom onset. And so by the time you have symptoms, you have amyloid deposition fully established in your brain, along with hypometabolism and significant atrophy that neurologists see in the clinic. So we have, as Dr. Sano beautifully pointed out, have made tremendous advances in the biomarkers in the last a couple of decades. So we have ways to look into brain MRI. And you can see here, this is the control brain. This is someone with mild cognitive impairment. You see atrophy here in the hippocampus. And someone with full-blown Alzheimer's will have a lot more atrophy in the hippocampus as well as in the neocortex. And we have ligands to measure amyloid in the PETs in the brain, ligands for tau PETs, Dr. Haddon is going to talk about. We have ways to look into metabolism in the brain through glucose uptake, because the synapses in neurons, when become dysfunctional, you have less of uptake of glucose, which is the main source of energy in the brain. So besides amyloid PET, tau PET, MRI, you also have spinal fluid biomarkers in the brain. And more recently, as Dr. Sano pointed out, we have plasma. And I was being asked to talk a little bit more about plasma, which I'm going to delve a little bit more. But when to order each of those tests, it really depends. For example, for a participant for a patient who is suspecting something inflammatory or autoimmune, then you end up doing spinal fluid through spinal tap versus someone who is going for one of those anti-amyloid therapies that Dr. Sano pointed out. You want to measure how the amyloid was at the beginning, and at the end of treatment, the amyloid PET may be a better suit. So it really goes by each case. But as very important, those tests have to be done in taking into consideration the clinical context. So we really need to know the history of the patient very well, the cognitive data, the brain MRI structures, and then you have all those other extra molecular data that you have not only through PET scans as well as blood biomarkers. So really, as a neurologist, you have to put all this data integrated so that you can make the best decision for patients. So what is the appropriate use clinically? It is different than research for those blood biomarkers. So, patients would have objective evidence of cognitive impairment. And as I pointed out, this test has to be put into context for the clinical context. As Dr. Sano pointed out, just because you have a positive amyloid doesn't mean you have Alzheimer's. You could have symptoms of Lewy body disease and Alzheimer's is a co-pathology, or frontotemporal degeneration and Alzheimer's is co-pathology. We have to really put all this data into context to see what is truly driving the disease. And when do you do those blood biomarkers? When Alzheimer's is a suspected cause of the cognitive impairment, and those tests are expected to change the course or clinical decision-making, for example, improving diagnostic accuracy and determining eligibility for treatments, for example, triaging for the anti-amyloid therapies. Usually, we don't clinically use for asymptomatic patients because it could have a low positive predictive value. And right now, we don't have those anti-amyloid therapies on the trial. And I think many of you are helping us advance the field to understand if removing amyloid before symptoms start, if that could change the course or delay the onset of cognitive impairment. So we are eagerly awaiting for the results of the trials, which could change the clinical recommendations for those blood tests. So why are blood biomarkers exciting? Because it's easy access to the blood. I think everybody would rather have a blood test than a spinal tap. And you can have potential scalability, less expensive, more molecular data for clinicians. You can screen much faster patients for eligible for anti-amyloid, and then do the more sophisticated quantitative data such as amyloid PET. And we can much better triage and identify the right patients for which clinical trial. If you think about over a decade ago, the initial anti-amyloid therapies, we didn't have those biomarkers. So they are testing out therapies against amyloid, but 30% of the patients didn't have amyloid to begin with. So now we made tremendous advances in trying to understand what is the molecular driver, or at least the pathology data, so that you can identify the right patient to the right clinical trial. There's still challenges with blood biomarkers. We are learning. It's just you're beginning to put this in clinical practice in the real world. It's not good enough to monitor the anti-amyloid treatment response. It's much more complicated to translate the general data from a group of hundreds of patients to the individual patient, and you're looking one-on-one in front of you. So, and there are a lot of nuances coming out and different tests with different sensitivity and specificity. So we're really trying to engage, as Dr. Sewell pointed out, physicians in understanding better which test is best for each patient. And we need much better biomarker. Thanks for donating your blood, because we really need biomarkers for all the pathologies. We don't have great biomarkers in the blood for synuclein for Lewy body. We're starting to have in spinal fluid. We don't have yet for TDP-43. We don't have biomarkers yet that predict who is going to develop the ARIA, the swelling, or and those dangerous things with anti-amyloid therapy. So we have made great progress, but there's way to go, and we appreciate your engagement in the research. As Dr. Sano pointed out, we know now the spatial temporal progression, how those biomarkers progress, and those pathologies start, as I pointed out, 15 years before any symptoms start. And we're really getting prepared to treat patients as early as possible, as soon as you get more effective therapies for pre-symptomatic patients. But in the meantime, you can always keep all those measures that Dr. Sano pointed out, exercise and vascular risk factor controls of diabetes, hypertension, because all of those things are very important for brain health. And as I pointed out as well, then a lot of biomarkers that we are studying in terms of synaptic measures to see how patients progress, who is going to progress faster versus slower. Those are still under development. We now have the synuclein for Lewy body through spinal tap and skin. We still need to develop better of those tests for the blood, and TDP-43 and many others. So we are going to make in the coming years great advancements, hopefully, in better biomarkers in the plasma. I was part of this work group that published the criteria recommendations for use of blood-based biomarkers in clinic. And as Dr. Sano pointed out, we are using two cutoff approach to confirm the patients who have very clear positive versus very clear negative, and also have this intermediate zone that need for the testing with PET or spinal fluids. So there are some barriers for blood tests, but there is a lot of opportunities going forward, and we appreciate your engagement in helping us identify better biomarkers for non-Alzheimer's pathology, for ARIA. Now, the best one for amyloid is what we call phospho-tau217, but it would just come out with another biomarker called MTBR, which correlates better with the tangles that Dr. Heather is going to talk about. And we are starting to understand how using those biomarkers in combination can better diagnose and predict cognitive decline. And I, again, thank you so much for your engagement, and I'm happy to answer clinical questions, including about the anti-amyloid therapies that are relatively new. There's one question here. Is that going to be at the end or no? Okay, at the end, yeah. Maybe hold up your question a little bit. Thank you. And thanks for the whole team here.
Thank you, Dr. Pereira. Now we're going to hear from Dr. Hedden. Sorry, the slide table is so kind of awkward. As you can.
All right. Are you able to hear me? Great. Okay, so I've titled this slide, I'm Trey Hedden. I'm the core lead of what's called the Neuroimaging Biomarker Core of the ADRC here. And so everything I do involves imaging in some way, MRI and PET primarily. I've titled this slide Dedicated because, one, thank you to all of you and our dedicated participants who make this research possible, but it's also dedicated to a particular patient that I'm going to talk to you about. I'm going to make this personal, I'll bring it down to a single person and talk about their case. This is a case study of a participant who was in the ADRC. She was female, had relatively rare because she had 2 copies of the ApoE4 allele. This is the allele that is the largest genetic contribution that we know of to Alzheimer's disease, and having 2 copies, only about 2.5% to 3% of people have 2 copies of that allele. But it drastically increases the likelihood that you'll get Alzheimer's disease sometime during your life. When we measured her blood plasma, we found evidence for elevated phosphorylated tau, indicating she likely had tangles, elevated amyloid, and this marker called NFL, which is a marker of synaptic dysfunction. She entered the study relatively young at age 57, already had a diagnosis of mild cognitive impairment, so was already exhibiting symptoms, and began her research imaging at that time. She returned at age 59, 2.5 years later. Additional imaging was done at that time, already had progressed to a diagnosis of AD. We followed her cognitively for 5 years. She showed a rapid decline in her cognition, especially memory, over that 5 years. And then when she unfortunately passed away at age 61, her husband and she made the decision to donate her brain. And that is now in the careful hands of Dr. Creary's team, and we're going to continue learning from that. This is her amyloid PET scan, overlaid on an MRI, taken at the exact same time on our special scanner here that we call a PET-MR combination scanner. We can get 2 scans in one. And at session 1, you see that there is some evidence, especially on this midline slice, of elevated amyloid in the frontal lobes and in the parietal lobes. This is not super elevated amyloid, but it's definitely present. The interesting thing is that when we measured her 2.5 years later, it's almost identical. And in fact, when we quantify it, we found no change. The values were exactly the same on the quantification. So no change in amyloid over that time. Yet she had this extreme decline in memory symptoms. Her tau PET MR shows a different story. Okay, at session 1, she was clearly elevated. It was especially lateralized, but you can see evidence of the neurofibrillary tangles on this tau scan. There was also some evidence of atrophy in the mediotemporal lobe, the hippocampus, which is the memory center of the brain. And at session 2, 2.5 years later, all of that was increased. That tau went up, and the atrophy also showed was increased, or a decline in the volume of the hippocampus. You can also see some enlargement of the ventricles, which indicates more global loss of gray matter in the brain. And when we quantify that, there was a 14% increase over that 2.5 years in the tau levels, and a 17% decrease in the hippocampus volume. Now, again, this person donated their brain, and that we'll be able to correlate that with what we saw during life and understand at the molecular level from those brain slices how the neuropathology was present in the brain at that time. Now, I want each of you to think about whether you have participated in imaging or not. If you've had a brain scan here at the center, a research brain scan, an amyloid PET, a tau PET, an MRI, it's likely that you're one of the dots on this screen, okay? This is actual data from our participants. And what I've done is split it up into the 3 different primary dimensions that we look at in characterizing Alzheimer's disease from imaging. That's amyloid PET, tau PET, and hippocampal volume, okay, on the MRI. 2 things I want you to take away are that when you look at the relationship between amyloid and tau, there's almost nobody in this upper left quadrant. If you don't have evidence of elevated amyloid, it's very unlikely that you have elevated tau, okay? Tau, as we saw in those curves that Dr. Pereira and Dr. Sano showed us, the tau follows the amyloid. Okay, we don't know that there's a direct causal link, but it seems to come afterwards in the progression. And the second thing is, although the hippocampal volume is decreasing along with amyloid, it's actually much more strongly related to the tau levels. So again, there's very few people in this quadrant, the upper right quadrant of this lower graph, indicating that it's rare to see drastically reduced hippocampal volume in the absence of tau. So it's very likely that the tau, which is inside the neurons, the amyloid plaques are outside the neurons, the tau tangles are inside the neurons. They're damaging those neurons, causing dysfunction and causing the hippocampal volume to decrease. And that, again, is the primary memory center of the brain. We can also track memory change over time. So we have participants who have been with us for at least 5 years, and we test them every year annually on a set of memory tests. And if you show low amyloid, this green line shows that if you have low amyloid, you stay relatively stable or even increase due to those practice effects that Dr. Sano was talking about. You're getting the same test over and over, you get better at it. And so if you have low amyloid, you tend to be stable or increase over time. If you have high amyloid but low tau, that's this orange line, you start off a little bit worse, but you actually continue to benefit from those practice effects. If you have high amyloid and high tau, it's a different story. You start off lower, you might get a small bump in the early years, but you tend to decrease over time. Okay? So again, it seems that the tau is driving that memory decline more likely than the amyloid. So even though our treatments are directed at amyloid, and that is the way we define the disease, there is this other primary hallmark pathology of tau tangles, and that is much more likely to be associated with the hippocampal volume loss and the memory decline. Okay, so what do we know and how do we learn more? So again, just to reiterate, it's unlikely that you're going to see extensive tau without elevated amyloid. Tau is more likely to drive memory change than amyloid. The amyloid therapies that Dr. Sano was talking about are remarkably effective at reducing amyloid plaques. I am just amazed at the way that they can reduce it. It's incredible. But they have a small impact on memory in the short term. So the studies do find significant effects. You see that people on the therapies are still going down, and the effects are small enough that they may not be detectable in a single individual. What don't we know? So does reducing amyloid early on prevent tau accumulation and long-term memory change? If we don't see tau in people who don't have amyloid, if we get rid of the amyloid, will they ever get tau? We don't know. We need to study and follow people for a long time to figure that out. And will it stop that memory change that I showed you on the previous slide? Again, we don't know. The field is moving towards a prevention model, exactly as Dr. Sano described. I think this is where we need to go. I believe we can get to a world where nobody ever has to experience memory loss from Alzheimer's disease again, but it will be by preventing it. And the more you partner with us, the more we can learn. Okay? Everything that you do, everything that you give, it helps us learn more. Everybody you see wearing a black t-shirt here today or a white t-shirt here today is basically employed to help you understand your brain. Okay? Thank you for giving us jobs, and thank you for making the world a better place. So how can you get involved if you're not already involved, or if you want to get more involved, especially in the imaging side? The imaging is expensive. We make a significant multi-$1,000 investment in people who have the imaging done. It's currently the only way to know where AD pathology resides during life. These blood tests are amazing. I can't believe, just 10 years ago, I would never have thought that it was possible to detect these in the blood. It's in the brain. How can it be out so obviously in the blood? But you don't know where it is. You can know that it's elevated, but you don't know where it is in the brain. And in that particular case I showed you earlier, we could tell that she had elevated amyloid, but we didn't know that it wasn't that elevated and that it didn't really change over time. So only with the imaging could we see that. For you personally, it provides an important baseline for detecting later change. Whether that's on the amyloid scan, the tau scan, or even just an MRI, it helps us understand where is your brain now so that we can figure out how is it changing over time. And one thing I want to point out is that the research scans that we do are more detailed and more comprehensive than clinical scans. I'm going to mention one study that uses a special scanner called the 7T that does some really in-depth, very high resolution scans. So if you just get an MRI from your doctor, yes, that's great, but it's not as detailed as what we do in research. So please consider participating in research as well. We have many imaging studies in addition to all the clinical trials that Dr. Sano mentioned, some of which involve imaging. We have a large nationwide study that we're involved in called the Consortium for Clarity, trying to understand Alzheimer's disease through blood, tau, PET, amyloid PET, MR, and FDG PET. One of our panel members is a member of that study. We have what I call our brain stars. These are the people, the most dedicated research participants who have done all of these different types of imaging. And then we have this ultra high resolution MR study. You can get more information about that study at the table outside. It also is looking at the gut microbiome and how diet and your microbiome affect cognition. I put our contact information down at the bottom. Feel free to take a picture of that and let us know if you're interested. And we also have at the table outside, any of our coordinators or myself will also be out there, and we'll be happy to talk to you then. Thank you.
Thank you, Dr. Hedden. Okay, we have one more speaker before we talk together as a group. We have Dr. John Query.
Okay.
Lean in a little bit.
So I'm John Crary. I'm a neuropathologist, and I'm the director of the Neuropathology Brain Bank and Research Corps. And I too am extremely grateful, deeply grateful for your participation. See what I do here. For your participation in our program. There's so many opportunities to learn, but I wanted to start here. These are some pictures of the brain drawn by Santiago Ramón y Cajal, who was a Spanish neuropathologist, won the Nobel Prize for discovering the synapse. And he was the first person to show the beauty of the brain. To me, it is nature's masterpiece. It's extremely pretty. If you see these neurons here, they look like trees in winter. It really shows how delicate it is. It's its beauty and delicacy that makes it vulnerable to neurodegenerative diseases. There's a lot of different diseases that can affect the brain: Alzheimer's, Parkinson's, Lewy body disease, vascular disease, and the list goes on and on. As a neuropathologist, it's my job in the medical laboratories not only to diagnose these different brain diseases, but to also try to understand their mechanisms. By being able to diagnose neurodegenerative disease better, we're going to make our clinical trials better, and it's so, so important. We have these fantastic biomarkers that we've been talking about today, but we need to do more work. Number one, there's a lot of false positives and false negatives. It's too many false positives. We don't want to be treating people that don't need to be treated, and we don't want to miss people who should be treated, especially at the earliest stages of the disease where the pathology is the most subtle. So we think there's an enormous potential to continue to understand neurodegenerative disease by studying our patients. And that's why I chose to become a neuropathologist and to run the brain bank. So we have a table outside, and you can drop by and you can talk to our coordinators and our managers and learn about what we do. But you may be wondering, like, what is a brain bank and what do we do? Well, this is a program giving patients an opportunity to participate in what I think is one of the most meaningful ways to dementia research. By integrating the clinical results, the genetics, the biomarkers, and the neuroimaging all around a single patient, we have the best opportunity, we think, to understand the mechanisms of neurodegeneration. Basically, this is how our workflow is. We work with patients and families. We work with funeral directors for brain donation. The tissue comes to our laboratory. All of our teams of scientists and students study the brain tissue. We archive it, we freeze tissue, and then we distribute it to investigators, not only at Mount Sinai, but all over the world. So really, our mission is threefold. We have a clinical mission. We believe that studying patients, that really the truth, what really is going on, it resides in the patient, it resides in their tissue, and that every patient is different. We have a saying in the brain bank, every brain tells a story. Not everybody who has the symptoms, and I can't emphasize this enough, not everybody who has the symptoms of dementia has plaques and tangles that define Alzheimer's disease, and that we're extremely frustrated because they can overlap in the same patient at the same time. So somebody may have cognitive impairment due to multiple pathologies, co-pathologies, which makes the clinical trials even more difficult. We love to teach. We teach medical students, we teach postdocs, graduate students, interns. People come from all over to study these brains with us, and the doors are open to the brain bank, and some of you may have signed up for a brain bank tour, laboratory tour during the break. So you're going to get a chance to see a little bit more about what we do. But we think that training amplifies our impact. The more people we can get involved in the community, the more eyeballs we can get on these cases, the higher the chances we believe that somebody's going to come up with the cure. And also research, the more tissue that we can share, the more chances there are that somebody's going to come up with a great idea. So this isn't a brain bank where we count our success by how many pieces of tissue we have in the brain bank. We measure our success by how much we share. So I'm just going to tell you a little bit about how we're using the tissue and how we're studying the tissues right now. We're really at an inflection point in research and medicine. Really, the world is grappling with artificial intelligence and how that's going to impact our lives. There's going to be a lot of bad stuff, but potentially great stuff. And one of the things that we really want to do is use AI to study Alzheimer's disease. And I think that's one of the greatest opportunities. All medicine is going to be transformed by AI, but the parts of medicine that are very data rich, especially neuroimaging, MRI imaging, but also imaging of the brain tissues, is where I see the real potential. You may not know this, but we spend a lot of time looking at slides. And here's a picture of a drawer with slides of our brain tissue that's stored in a warehouse in New Jersey. Just tons and tons and tons of data, which is locked away on those glass slides. What we've learned how to do with new technology is to digitize all of these slides. So we're getting rid of the microscope. There's a microscope I have there on my desk. I don't use that. I barely look in my microscope now because all of my glass slides are digitized now. And that is extremely efficient. It's great because I can easily review cases and I can share the cases with people, but it gives us the opportunity to use computer vision to study the brains. Now, when I describe looking at the human brain to people, I usually use analogies to explorers. It's a lot like being dropped off in another continent or the bottom of the ocean, or maybe even better, like on another planet, right? And you're walking around and trying to understand what all these things are. We've come to understand what a plaque looks like and a tangle looks like, and we correlate, but there's so many different types of cells. There's so many different types of changes. There's so many different types of states and connectivity. The brain is really the most complicated organ. I think it's probably the most complicated machine that we've ever encountered ever in the known universe. So the opportunity of being able to train an AI algorithm to go through our brains and to measure all of the different structures all at the same time, and then to correlate with different outcomes is enormous. So I usually use the analogy of like a needle in a haystack. Right now, I had to, up until recently, to go in and count all the plaques and the tangles to measure them, and then we could correlate them with the PET scan and the biomarkers and see if they were statistically correlated. And that's how we did our research. Moving to AI, I can have that done automatically. It's great because it's rigorous and reproducible. It's like looking for a needle in a haystack where I can just run the AI and look at the haystack. But What I've learned with the most recent algorithms is we don't have to tell it what to look for. You may say, hey, here's a haystack. What's wrong with it? It may find the needle, but it could also find other things like a mouse's nest. And you may say, oh my gosh, I didn't think there was going to be a mouse in this haystack. Or, the species of plant that makes this haystack is different from this haystack. And you say, oh my gosh, I can see that. Actually, the AI algorithms can also tell you that there's something wrong with the haystack, but it's so multidimensional and complicated that our human brains can't understand it. But it's still something different about this haystack. So imagine now us with powerful computers, Mount Sinai supercomputer Minerva, we're able to go through all of these tissues that are donated to us and discover new features, things that we never knew were there, things that we were never capable of measuring before. This is what we're doing now at Mount Sinai with the brain bank. Here, that's just a picture of a hippocampus in the upper right, which is the memory center of the brain, with an AI overlay of the different regions. It's a heat map, the different regions that are most strongly correlated with cognitive impairment. And we can go in and look at the cells here and map all of them across the entire brain. We see enormous potential using this approach. Let me give you an example. Age estimation. We're really interested in brain health, which has a lot of different components to it. Being able to estimate somebody's age is really important because you can see if they have accelerated aging. Are they older than they should be chronologically? Biological aging and chronological age are different. Or maybe they're younger than they should be chronologically. Why does that happen? Genetics, environment, maybe response to a drug treatment. This is really important, but to be able to put a number on your biological age is really difficult. And as a neuropathologist, I can't just look at a slide and tell you how old somebody is. That's just not a task that can be done. We've trained an AI here at Mount Sinai that can estimate brain age within 2 years. We've also found that, for example, patients with repetitive concussions who have participated in contact sports can be accelerated up to 10 years. So these are patients with chronic traumatic encephalopathy. The AI can now give us this measure of 10 years on average acceleration. So this is extremely powerful. We're pulling out other signatures, psychiatric structures in the brain that are correlated with psychiatric illness we've never been able to detect before. What we're hoping to do across Mount Sinai is develop a learning health system. And I'm starting to reconceptualize the brain bank as a learning research system. What that means is that every single participant who comes into our program will instantly be added to the database and the models will be retrained. And this, when integrated across all the modalities, clinical data, genetic data, neuroimaging, biomarkers, it's going to give us a model, much like ChatGPT, but it would be a brain version of ChatGPT that we think we're going to be able to ask questions to, for example, given this environmental exposure, given this genome, given this scan, will this patient, for example, respond to a treatment or not? So this is where we're going. It's extremely exciting, and Mount Sinai is really leading the way here. And it's really because of you and the other participants that we're able to do these extremely exciting studies. Thank you very much.
Sure. So the most important people on this stage, ooh, that is really annoying. The most important people on this stage, not to diss any of you, are our two research participants here, Alex and Christiana. And I was hoping that you guys would each just say a little bit about what made you decide to participate in our research and what kind of research you've participated in.
There, better. Can you hear me?
Yeah.
Okay. My husband had a traumatic brain injury, which catapulted him immediately into loss of cognition and memory as if he had advanced Alzheimer's. And I took care of him at home for the last 10 years of his life. But before, when it first happened, we were at a great loss. what to do. And then we found this wonderful department at Mount Sinai that looks at brains, wants brains, and supports those of us who donate our brains, so that we turned something bad, which was my husband's accident, into something good. And we both were very glad about that. It became some purpose, in a sense, for his malady. The second thing is that once we donated our brains, and they assured me that they wanted normal brains too for comparison, every single year, Dr. Neugeschel and her people come to give me a 2 or 2.5 hour test of cognition and memory. And I love doing that because they give me the reports, and I'll know what's happening as I continue to age, if I'm lucky enough to do that. And then the third thing is that Dr. Neugeschel, here to my right, oh, I'm sorry. Yeah, okay, over there. She has made herself available to me for all kinds of questions about my own conditions and even other members of my family. And that is just so invaluable to me, and I thank you. So for me, having donated my brain, I have all of these benefits. And,
Plan to do.
oh, right. Well, I was signed up to donate my brain. Okay.
In some ways, Alex and I have a similar path, and you speak my mind.
Oh.
My husband was not in a traumatic accident. He had a gradual onset and came here to Mount Sinai seeking diagnosis. And his original diagnosis was a little confused. They weren't sure. It was one of 3 things. And we had to wait a little while to see which symptoms became dominant, and found that he had PSP, which is parasupernuclear palsy. It's much easier to say PSP. And I took care of him at home for 5 years after the symptoms became evident. And we found exactly the same thing that you found, Alex. And we found exactly the same comfort. And it gave our lives so much more meaning to say, at least we can share what we have, and maybe science can learn something more about it. And everything that everyone has done prior to this gave us so much comfort that people were able to speak to our condition and know where we were. And even though we didn't like it, that he had a condition that was completely untreatable, so comfort care was the answer. But even that was a huge comfort because people understood exactly where to meet us at each stage of this process. And then another unexpected comfort, I don't know if you found this to be so, but having agreed to donate our brains, and I'm the partner too, I knew exactly what to do when he died. And he died about 2 months ago. And it was wonderful to have conferred with the funeral home, to have spoken with people here, to know exactly what was going to happen. And then when the time came, it went very, very smoothly, and it was very beautiful and very comforting to everyone in our family. So I appreciate you all being here for us. Thank you.
Thank you so much. I also want to point out that both of you were actually decided to participate as well in our studies, in our
Yes,
yeah.
it's just as much fun. And I was very, reassured to hear that sometimes you get a little better at it.
She's in the image. And you're doing the imaging studies, right?
I don't
No.
even think about the names, just someone
I mean,
sent me an email saying,
excellent. Fantastic. So, I guess my first question, as I'm standing here next to the mic. So I think you guys have all really talked a lot about how each of your individual pieces are so important. And you've sort of shown how some of the connections happen. But I think something that people have asked me is, so if I have my blood test, why do I need to get a scan? If I have my scan, why do I need to donate my brain? And I'm just wondering whether people, anybody on the panel would like to address some of those questions.
Well, I'll be happy to speak to it first.
Sure.
I'm a visual learner, so I understand that people perceive things in many different ways. So the layers of study that you have going on makes perfect sense to me, because I think people learn differently. And the more minds that come and study this from the more complete perspectives, the richer the finding is going to be. And you'll be able to perhaps sometimes confirm and sometimes find a new road that you might not have thought going down. So I like the thoroughness. And it's also fun. I mean, everyone here is so nice. And, when you're going through this, it's not fun at all. And it's wonderful to meet people who have a goal, who have an objective, who can, who say, hey, this is a fallow field. This is, we like it that you're deteriorating. Not too many people say that to me,
So Dr. Hedden, you look like you have a thought.
Yeah. Well, so as I mentioned during the talk, that just having an elevated blood test tells you that you have the pathology. It doesn't tell you exactly which pathology necessarily, because some of the markers are actually associated both with amyloid and tau, and so you can't tell, do you have just one or do you have both? And the other is you cannot tell where it is. So if you care about where it is in the brain, and some symptoms are driven by the location of the pathology. So if you have tauopathy in a language center, you're more likely to have language symptoms. Right? So where it is in the brain can matter both to your symptomatology and to the diagnosis that you receive from the doctor, and might impact your course of treatment.
Yeah, I think those are all very important points for us as clinicians and for us doing research as well. It's critical to integrate all those pieces of data, right? Just one standalone blood test doesn't mean much. You really need to have the history, the brain imaging. For me, I look at all my patients' brain MRIs. I have some of my patients here. They can tell you, because where the atrophy is correlates in general with your symptoms are, and understanding of pathologies and blood tests. And ultimately for research, we really need those comparators. We could not have come with blood biomarkers without comparing to something. Something was amyloid PET scan, along with cognitive values. And ultimately, as Dr. Curry pointed out, the major truth is in the neuropathology, because we don't have biomarkers for everything right now. Hopefully, we're going to get in the future for TDP-43, beta-synuclein, and etc. But having this brain tissue is critical as well, to be able to understand mechanisms of disease, understanding the final diagnosis of the patients. I'm sure it was very important for you to understand the final diagnosis of your spouses through neuropathology. So having all this data together, it's critical for us to understand each patient individually, as well as to advance science and mechanisms of disease and biomarkers, etc.
Yeah, I would just add, just to make it real, the reason why the location is so important, the brain is very complex in the way that it's connected. And for diagnosis, understanding those brain regions is so critical. I describe it to people like the 3 things that are important for diagnosis, and it's the same as New York City real estate. Location, location, location. So that's what we're obsessed with. And if you look at the blood, it's like looking at the garbage that's coming out of somebody's house. You don't get to figure out which room it was from. You can guess, but we really are we're playing a game of Clue. Professor Plum with the candlestick in the conservatory. We really need to know, was it tau?
Was it in the, hippocampus? But that last part, how was it done? It was the rope, it was the candlestick, it was the gun. We want to know the mechanism. And we have an idea. We think that it's amyloid. And we've shown that we can get a little bit of improvement by removing the amyloid. But there's a lot of other stuff in there, and we don't know exactly what to target. And so that's why we really need the tissue. And that's why if we can profile it and if we can correlate with the symptoms, that we might be able to come up with some other really good ideas for treatment.
Yeah. Listening to what you're saying, it's really making me think that these are gifts that will continue to give over time, because as new ideas arise, having these tissues, having these samples will allow us to look through and think about new and different directions. And that's pretty exciting because obviously this field keeps growing and changing.
Yeah, I would just add to that. And actually, my own experience echoes your guys in a way that my father was in the brain tissue donation program and he died a couple of years ago. And you have his brain, I don't know, on a slide or in a computer or where it is now. But it's brought me a lot of comfort because it makes me think he's still living in a way. You can see that the contribution that he's making to science is going to go on for years and years and years. And that's been very meaningful to our family to know that. So I've had a similar experience.
If we're going to get personal, my father-in-law had dementia and he lived with that for over 10 years. And when we donated his brain, we found Lewy bodies and we had no idea that they were there. We were thinking that it was Alzheimer's disease. Actually, he had both. He had co-pathology. And so it turned out that he was fighting two diseases at the same time and we had no idea. So it does bring a lot of comfort, and it also shows in stark relief that we need to do a lot more in terms of diagnosing better. Because if we can't diagnose our patients well, it's going to make it really hard to design these clinical trials. We're making progress and we have some successful clinical trials, but we'll be able to test a lot more drugs at a lot faster, and the associations, the statistical power, our ability to prove that they work will be a lot stronger.
I want to add that my son has now signed up for brain donation too, because he sees how much I get from it. He wants to get that too.
Thank you for adding that.
I have a question. Is it okay if we?
We can take a question or two and then we're going to stop for lunch.
You sure don't.
How useful am I? Don't come into it at the beginning. Sign up for studies. But somewhere along the road, I get some epiphany. I said, I take the brain. Okay. But I've never done anything else. Am I useful to you as a participant in the program, in any phase of the program? Of their research protocols, looking to engage with me, but just on my brain only. Why am I brain out? To say that I don't come into it until the very end.
I mean, I can just say for a moment that it's the longer you're with us, the richer the data, the richer the information. And, it's nice for us because we get to know you, and that's an important thing. And it makes the whole experience much richer for us. However, we do have people who decide to donate usually it's their family members' brains, like right at the time of death. And then in that case, we just try to do as much historical digging as we can. And we spend a lot of time talking to family members, getting information from doctors as much as we can. So it is something that is a possibility. It's just, it's not our favorite thing, but it's good. It's still very
I'll just add to that too, that a lot of time people come into the ADRC who just want to be followed annually and get the cognitive testing every year. And then as the years go on, they may change their mind and say, you know, maybe I will join an imaging study, or maybe I will sign up for brain donation, or I'd like to be screened for this clinical trial. And that's the value of having this big group of people that we follow every year, because people change their mind and new studies start, and you're the first that we'll go to talk about it.
I have one.
Just as an example, not only as a cognitive neurologist, I also have a lab here. And all the questions I try to address in my lab are inspired by patients that I see in clinic, and vice versa. My ultimate goal and mission when I wake up and my daughter is here, Clara, is that makes me go to work and they tell mom, you've got to find a cure for Alzheimer's, is to try to find something to improve the health and prevent this terrible disease. But just to understand, for example, we leverage on the data sets from this Mount Sinai. We looked into, wanted to understand the question in clinic is, why do some patients progress very slow? Why others progress very fast? Just as an example, I usually give, there is a lady that I've been following for 7 years. She's amyloid positive, very amnestic. And I live on the west side. I still run into her into Zabar. She can still go shopping. She's a very slow progressor. As a contrast, there's a retired psychoanalyst presents the same symptoms. They were both MCI when I diagnosed them, amyloid positive. In 4 years, his wife had to put him in a retiring home because she couldn't take care of him in the house. So progress really fast. Why are some patients progress fast, others slow? So we came to Dr. Marisano and the neuropsychologist team and trying to see how many brains you have of patients with longitudinal cognitive data for the last multiple years. And you could track and make all those graphs of how fast patients progress. And you use those brains and you're trying to understand that we saw in vitro assays that how much this tau that we are talking about sitting, how much they propagate in vitro correspond or predict how fast patients progress in a particular brain area called inferior temporal gyres. And then you did a transcriptomic analysis to try to understand the drivers. Why is someone progressing fast or others slow? So just for you to understand how those questions that we have can be start off, we can address them by going to huge data sets like the Alzheimer's disease research data sets of brain and cognitive data that you do every year. You use every single point to be able to analyze this data set.
I'd like to speak to this question too, really quickly. It's all useful. And I always think about that cathedral in Cologne, Germany. It took them 600 years to build it. And this actually is close to where Alzheimer was born. Do you imagine you start this project and it's like, you'll never live to see them finish it. But you're putting these stones on and laying the groundwork and the foundation. I would think of it like that. Every single time you come in, you're putting a stone in and you're building them. And every time you do a scan, maybe some stones are bigger than others, but you're adding as much as you can. So we suck up that data, it goes into our models, and the more the better. So you can do what you can do, and we are extremely grateful. This is a long-term project. I hope that AI is going to accelerate it. It doesn't have to be 600 years to get to the cure for Alzheimer's disease and progressive supranuclear palsy and all those other dementias. We can accelerate that. So I'm very optimistic that in the next 10 years, we're going to be looking at a completely different landscape.
And from an emotional perspective, you cannot find a more personal legacy. It is unique to you.
She said from a personal point of view, you can't find a more meaningful legacy.
Yes, I had a question. Am I correct in understanding that the two drugs that you mentioned that have an effect on the amyloid do not have an effect on the tau tangles?
It hasn't really been directly tested for one of the drugs, and there was a secondary analysis on the other. Mary, do you remember the exact details of that?
So there's some evidence that by slowing the amyloid, you don't get as much of the tau, but it's not a treatment for tau. And in fact, that's the next protein that many of our studies are going after. We have a big study platform called the tau platform that will be studying many drugs at the same time. So we're very excited about expanding. So it only starts the process. And if you remember this slide, nobody got cured. They just got a little bit less deterioration. So there's so much more to do. But as you can see, we can do it when we do it together.
I'll just add that in animal, thanks, Mary, beautiful answer. I'll just add that in animal models, we know that the amyloid is critical to accelerate the tau propagation. So it may play a role in the biology of the tau propagation. As Dr. Haddon and Dr. Senna have pointed out, it's really when the tau gets out of the middle temporal lobe to the neocortex that the patients start having major cognitive impairment. And that's what we want to do next. It's much harder to target tau than amyloid because amyloid is outside the neurons, tau is inside the neurons, but there are major efforts in trying to do that. And ultimately, we may end up with multiple drugs, one against amyloid, one against tau, one for autophagy or inflammation, and understanding precision medicine better because there's a lot of heterogeneity in Alzheimer's disease. They are not all the same. Actually, each patient is a different story. You know, each brain is a different story. And that's why I go to clinic and each family is a different story. So we're trying to ultimately get to more precision medicine. Now, there's beautiful work by some groups, including ours, in terms of you look into the spinal fluid of the patients and subtyping. There's the molecular markers. So some patients are more inflammatory prone, other are more vascular prone, other are more excitable, and ultimately trying to, through the use of biomarkers, individualize more precision medicine in terms of therapies. That gentleman with glasses was asking for a question a long time. Yeah.
Is a very brief question.
Yeah.
And then we're going to stop for lunch. Can we get him the mic? Actually, I'm the closest.
Couple of points. But the first thing I want to do is really thank you so much for having this patient-centric conference. I think I speak for all of us when I thank you for that. The first point, my wife has posterior cortical atrophy. I have 4 boys, and I understand that the biomarkers are not recommended for people who are not symptomatic. But my question is there a utility in my children getting measured and then engaging in the activities? I didn't hear Dr. Sano's presentation, but there are many things that are associated with decreased risk. And if they have elevated biomarkers, wouldn't it make sense for them to pay attention to blood sugar and to exercise regularly? They're all so busy. It would be nice to say, hey, you know, you really need to exercise, for example. So I'd like you all to comment on the prophylaxis. And then number 2 is I understand that there is a model of amyloid and tau, and you guys have reinforced that over and over again. But I wonder if if you can comment on the model of inflammation equals amyloid equals tau, because as you're pointing out, it's multifactorial. It's different for everybody. The sources of inflammation are very numerous, and maybe that's one of the keys to the variability. So thank you for your comments.
First one, I'll just...
We'll divide up the question. Well, the good news for you, your family in particular, is that, so for the audience, posterior cortical atrophy is a variant of Alzheimer's. So patients start accumulating the tau pathology more posterior areas of the brain, called the parietal, or even the visual cortex, and the memory presents much later on. Unlike the typical form of Alzheimer's, it just starts with memory in the hippocampus and then goes to cortex. the good news for your family is that posterior cortical atrophy is much more sporadic genetically than the typical Alzheimer's. So knock on wood, maybe that was just your spouse. So the question on doing biomarkers on asymptomatic patients, there's a lot of controversy, and we're still trying to figure out what is best as a field. As Dr. Sano pointed out, there's been the Pointer study that did study patients at high risk and exercise, keeping socially engaged, keeping cognitively engaged, having a healthy diet, like the mind diet. All those things are important, just not only for the brain, but also for the heart. Okay, some people would argue we should be doing that anyways. Why do you need to do a biomarker? The problem in the field is that if we start doing right now biomarkers in individuals who are asymptomatic, first of all, there's going to be a huge amount of false positives and a lot of people worrying, and there's not a lot more we can do immediately. We always eagerly waiting for the results of those trials in which many of you may be engaged, which removes amyloid in pre-symptomatic patients and seeing if it's going to delay the onset of symptoms. We are still waiting for the results of those trials. So once you have those, that the recommendation may change. So right now, the recommendation for using those biomarkers clinically are for symptomatic patients for those reasons. But many of you engage in research protocols. You can get a lot of those data, including brain MRIs and many of the cognitive data, and you can detect as early as possible. And with the field that's been pointed out, has been moving forward for the earliest stages and even pre-symptomatic. And while in the last 10, 20 years, we made tremendous advances in biomarkers, as you pointed out, imaging, amyloid PET, tau PET, CSF, now blood. Now we hope in the next decade, we're going to advance in therapy at the earliest stages of the disease, because we can diagnose molecularly those patients way before they have symptoms. And that's where we're moving forward.
Thank you, Dr. Pereira. I'm going to pause the rest of the responses to these great questions until 2:00, when we're going to have a whole Q&A panel to take everyone's questions, and we can continue talking to them, because I'm aware lunch is here. I want to pause and let everyone stretch and get something to eat. So we have lunch outside on several tables. Everyone will be out there to help you. If you have any mobility issues, or you'd prefer to stay seated and have someone bring you something, just let us know, and we'd be happy to do that. And then you'll come back in here and have some lunch and hang out, and then we'll talk about the breakout groups after lunch. And well, I'm gonna wait till everyone's back in the room. All righty, thank you so much. Let's eat.